So, good morning, everyone. Our topic for today is all about the brief history of biochemistry and the introduction of biochemistry. Our learning outcomes for today's output is to know and understand the history of biochemistry, to define biochemistry and its importance, to determine the four categories of biomolecule and its role, and to determine the parts and function of plant cells. So, a quick brief history of Earth and life. So, Earth was formed about 5 billion years ago when our solar system took shape around the sun. As you have seen in the picture, we have here the stargazing light. So, it represents the universe throughout of time. So, because of Big Bang Theory, the universe has expanded and cooled ever since. There is an inflation. There is a high energy particle reaction. So the first nuclei was formed a few minutes and 3,000 years. The first atom was being formed. So the few 100 million years, the first galaxies and stars are being formed, and a few billion years, the expansion of the universe begins to accelerate. So, a current model for the origin of life on Earth is the bubble hypothesis. The origin of life is still a mystery, but there are some theories that speculate how inorganic matter was transformed into the organic matter that created life. In 1992, it was proposed that bubbles in the sea were the key that helps to create complex organic matter that eventually became life. So here is the schema of the bubble hypothesis. So there is a volcano erupted under the sea, releasing gases enclosed in bubbles. Second, the gases considered concentrated inside the bubbles reacted to produce a simple organic molecules. When the bubbles persisted long enough to rise to surface, they pop, releasing their content to the air. And because the sun is being bombarded by ultraviolet radiation, lightning, and other energy sources, the simple organic molecule released from bubbles reacted to form more complex organic molecule, and the more complex organic molecule fell back in the sea in raindrops. When there when there they could again be enclosed in bubbles and begin the process again. So the first cell are thought to have arisen from aggregation of molecules that were more stable and therefore persisted a longer. It has been suggested that RNA may have arisen before cells and subsequently became packaged within membrane, which is we call the RNA world. So bacteria were the only life form on Earth for about 1 billion years. This is the first methane utilizer that being used. Then anaerobic photosynthesizers are also known as anoxygen, so sorry, an or oxygenic photosynthesis is the process by which a certain bacteria use light energy to create organic compounds but do not produce oxygen, and eventually auto-forming photosynthesizers. So anaerobes are those bacteria that cannot use oxygen to generate energy. Oxygen levels rise about 2.5 billion years ago. This is thought to have been created by photosynthesis by cyanobacteria leaving the sea. The oxygen-rich ozone layer was also established, shielding the Earth's surface from harmful solar radiation. This further provided our planet for appearance of inhibition by other organisms, including us. Fossil indicate our own species, Homo sapiens, arose in eastern Africa some 190 years ago, first venturing beyond Africa about 70,000 years ago. The first eukaryotes can be seen in the fossil record about 1.5 billion years ago. All organisms other than bacteria are descendants. So now we will talk about the brief history of biochemistry. So Antoine Lavoisier or Antoine Lauret de Lavoisier was born on August 26, year 1743, and he died in May 8, 1794. He is known for combustion, identified oxygen, identified he identified hydrogen, stoichiometry. He discovered oxygen, carbon, nitrogen, and silicon. When we say combustion, this is the process in which substance react with oxygen to give heat and light. So we have also in part of the brief history of biochemistry, we have also the vitalism. Until the early 1880s, vitalism was a common belief. The compounds found in a living organism can only be produced by living organism and could not be produced in laboratory. Now, an obsolete scientific doctrine, vitalists argued that it was the presence of a vital force. When you say vital force, this is the life force or spirit that distinguished the living organic world from inanimate organic inorganic world. So when we say organic molecules, these are molecules of life, and these are built around chains of carbon atoms that are often quietly long. There are four main groups of organic molecules that combine to the cell and their parts, and these are the carbohydrates, proteins, lipids, and nucleic acid. We have also Frederick Wöhler. He disapproved the belief of vitalism in 1828 by synthesizing urea. When you say urea, this is an organic molecule and a waste product of animal metabolism. This is a colorless, crystalline compound which is the main nitrogenous breakdown product of protein metabolism in mammal and excreted in urine. He showed that heating silver cyanide, an inorganic compound, and ammonium chloride, another inorganic compound, produced urea. Without the aid of a living organism or part of a living organism. Note that many science historians believe in this vitro synthesis of urea by Wöhler as the starting point of biochemistry. We have, however, many consider Edward Buchner's first demonstration of alcoholic fermentation in 1893 in a cell-free yeast extract as the starting point for the birth of biochemistry. This was another blow to the vitalistic thinking, showing that the presence of living yeast cells were not needed for fermentation. Previously, scientists believed that only living cells could catalyze such complex biological reaction. Pasteur was the first to demonstrate experimentally that fermented beverages result from the action of living yeast, transforming glucose into ethanol. Moreover, Pasteur demonstrated that only microorganisms are capable of converting sugars into alcohol from grape juice and that the process occurs in the absence of oxygen. Edward Buchner had shown that fermentation could occur in a yeast extract free of cell, making it possible to study fermentation biochemistry in vitro. He prepared cell-free extract by carefully grinding yeast cell with a pestle and mortar. The resulting moist mixture was put through a press to obtain a juice, to which a sugar was added. Using microscope, Buchner confirmed that there were no living yeast cells in the extract. Buchner detected zymase or enzymes, the active constituent of the extracts that carries out fermentation. He realized that the chemical reactions responsible for fermentation were occurring inside the yeast. So Pasteur was the first to demonstrate experimentally that fermented beverages result from the action of living yeast, transforming glucose into ethanol. Moreover, Pasteur demonstrated that only microorganisms are capable of converting sugars into alcohol from grape juice and that the process occurs in the absence of oxygen. Edward Buchner had shown that fermentation could occur in a yeast extract free of cell, making it possible to study fermentation biochemistry in vitro. He prepared cell-free extract by carefully grinding yeast cell with a pestle and mortar. The resulting moist mixture was put through a press to obtain a juice, to which a sugar was added. Using microscope, Buchner confirmed that there were no living yeast cells in the extract. Buchner detected zymase or enzymes, the active constituent of the extracts that carries out fermentation. He realized that the chemical reactions responsible for fermentation were occurring inside the yeast. We have also the Miller-Urey experiment, which experiments recreating atmosphere of primitive Earth with the energy sources and temperatures have led to the spontaneous formation of amino acids and other biologically significant molecules. The Miller-Urey experiment showed that a variety of organic molecules, including the amino acid, could form in an early reducing atmosphere. Living things obey the standard law of physics and chemistry, and no vitalistic force is required to explain life at the molecular level. So in 1810s and 1830s, a major substance from animals and plants were identified. It composed of carbon, hydrogen, oxygen, and nitrogen. The term protein, meaning the most important thing, was first used in 1838. In 1850s to 1890s, carbohydrates, lipids, and nucleic acid were recognized. The term biochemistry was coined in 1870s and the term biology in 1880s. The question is, what is biochemistry? Biochemistry is the study of molecular basis of life or understanding life phenomena in chemical terms. The main focus of biochemistry is to understand how biological molecules give rise to the processes that occur within living cell and whole organisms. Biochemistry is closely related to molecular biology because molecular biology is the branch of biology that deals with the structure and function of the macromolecules that essential to human life. The study of molecular mechanism by genetic information encoded in DNA is able to result in the process of life. So when you say genetic, this can be defined as a study of genes at all levels, including the way in which they act in the cell and the cell ways in which they are transmitted from parents to offspring. The question is, what is biochemistry? Biochemistry is a study of molecular basis of life or understanding life phenomena in chemical terms. The main focus of biochemistry is to understand how biological molecules give rise to the processes that occur within living cell and whole organisms. Biochemistry is closely related to molecular biology because molecular biology is the branch of biology that deals with the structure and function of the macromolecules that essential to human life. The study of molecular mechanism by genetic information encoded in DNA is able to result in the process of life. So when you say genetic, this can be defined as a study of genes at all levels, including the way in which they act in the cell and the cell ways in which they are transmitted from parents to offspring. So we have four important biomolecules that we need to remember. So the first one is the lipids, second is the nucleic acids, the third one is the carbohydrates, and the fourth one is the proteins. So what does biochemistry study? So biochemistry studies all the processes within living cell, with their all molecular compounds and contents, understanding how all these processes contribute to health and diseases of living cells or organisms are major focus of biochemistry. So we have here a diagram showing how biochemistry relate to others. So we have genes, and genes is it has protein, and protein that studies biochemistry. And the function of gene is to provide a genetic information in a human body. So in the center, we have see a recombinant DNA. So, the chemical basis of life. We know that the life itself is constructed from non-living matter. When we say non-living matter, these are atoms and elements. Living organisms have about 20 elements compared to 94 elements occurring naturally. We have carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, which is known as chomps. Chomps is a mnemonics for us to easily memorize those six elements. Account for more than 97 of the weight of the most organism. So, the relative amounts of these elements may vary among organisms. Water is a major component of cells and accounts for a high percentage by weight of oxygen. Carbon is much more abundant in living organisms than in the rest of the universe. Some elements such as silicon, aluminum, and iron are very common in the Earth's crust but are present only in the trace amounts in cell. So we have here a periodic table. So there are colors that being indicated. So the important elements shown in the living cell are being shown in it. So here in the we have the red elements. The knobs are the six abundant elements, and these are the carbon, nitrogen, oxygen, silicon, aluminum, and iron. The five essential ions are the purple ones. So the sodium, magnesium, calcium, potassium, and chlorine. So the trace elements are shown in dark blue or the more common, and the light blue are the less common. There are two major breakouts in the history of biochemistry are especially notable. So the first one is the discovery of the roles of enzymes as catalyst. So the experiment of Butcher showing that extract of yeast cell could catalyze the fermentation of the sugar glucose to alcohol and carbon dioxide. So we have enzyme, there is a substrate, and then when the enzyme and substrate have a complex, it can produce product. So the second one is the role of nucleic acid as information carrying molecules. So DNA, excuse me, DNA is the genetic material in 1940s. The three-dimensional structure of DNA, the DNA double helix, is being studied in 1950s, the genetic code in 1960s, biological information like central dogma of molecular biology in 1970s, manipulation of DNA, catalytic activity of RNA or also known as ribosome, the cause of cancer, genomic era, brain research in 2010s. The importance of biochemistry. What is why we are studying biochemistry? So using biochemistry, it makes us aware in determining how much medicine should be prescribed or given to patient. It ensures that nobody gets too high of a dose for their body and that people recover is in a little time as it possible. So the nutrients value of food material can also be determined by biochemical tests. So bio means life. Chemistry talks about how things interact. So when we say biochemistry, this is the branch of science in which you study the chemical and physical processes that occur in an organism. The science that is concerned with the structure, interaction, and transformations of biological molecules. That is the chemistry of life. Chemistry can be subdivided in three principal areas. So the first one is the structural chemistry. The second one is the metabolism. The third one is the chemistry of processes and substances that stores and transmit biological information, or also known as molecular genetics. So the cell is the fundamental unit of life. Prokaryotes and eukaryotes, we have that both. And in eukaryotic cells, we have animal cell and plant cells, where in the plant cells, they have chloroplast and cell walls. Cells are composed of small molecules, macromolecules, and organelles. So you have seen in the diagram or in the screen the approximate chemical composition of bacteria cell. So we have here the water, inorganic ion, sugar, precursors, amino acids and precursors, nucleotides and precursors, lipids and other molecules or macromolecule. So in this screen we have seen the percent of total cell weight and the number of types of each molecule. Except for water, the of most of the molecules found in the cell are macromolecules, and it can be classified into different categories. So we have here the lipids, carbohydrates, the example is starch, the proteins, and the nucleic acid, which is the DNA and RNA. Lipids are primarily hydrocarbon structures. Carbohydrates, like lipids, contain a carbon backbone, but they also contain many polar hydroxyl groups and therefore very soluble in water. Proteins are the most complex macromolecules in the cell. They are composed of linear polymers called polypeptides, which contain amino acids connected by peptide bonds. each amino acid contains a central carbon atom attached to four substituents. So, the four substituents are a carboxyl group, an amino group, a hydrogen group, a hydrogen atom, and an R group. Nucleic acids are the large macromolecules in the cells. They are very long linear polymers called polynucleotides, which is composed of nucleotides. So this is the structure of lipid. This is the structure of carbohydrates. This is the structure of amino acid. And this is the structure of DNA contained four bases: the adenine, cytosine, guanine, and thymine. So this is have you seen in the diagram, this is the covalent structure of DNA. So a nucleotide contains a 5-carbon sugar molecules. One or more phosphate group is a nitrogenous base. So in DNA, we have adenine, thymine, guanine, and cytosine. So the 4 RNA bases are adenine, uracil, guanine, and cytosine, often referred to us as AUG and C. The biochemical energy. All cellular functions required energy. The most important chemical form energy in the most cell is ATP. ATP, ADP plus PI. Most ATP synthesis occurs in chloroplast and mitochondria. When we say ATP, this is adenosine triphosphate. The ATP is considered by biologists to be the energy currency of life. So it has the high energy molecule that stores the energy we need to do just about everything we do. So we have here the energy transfer, the photosynthesis, and the ATP, where it happened. And next one, we have the transfer of information from DNA to protein. So before we proceed to the plant cells, I would like to clarify that this presentation is only an introduction for us to be informed and not to be surprised in encountering those words which is required for us to understand the next presentations. So now let's move on to the plant cells. So the plants are Earth's primary producers. Harvest energy from sunlight by converting light energy into chemical energy. They store this chemical energy in bonds formed when they synthesize carbohydrates from carbon dioxide and water. So non-motile have evolved to grow towards resources throughout their lifespan. The vegetative body consists of a leaf, which is for photosynthesis, a stem for support, a roots that anchorage and absorption of water and minerals, the nodes, which is for the leaf attached to the stem, and the internode region of stem between the two nodes. So we have here the leaf structure, and as you have seen, there is a layers, the same as human body. So we have also the stem. So there is also a layer. So these are the parts of it. The roots. So this is the full structure of the plant cell. So there are a lot of parts of it, and they have the different function. First, we have nucleus. It contains almost all of the genetic materials. What is contained is called the nuclear genome. This varies greatly between plants or species. Surrounded by nuclear envelope, which is doubled membrane, same as plasma membrane. The nuclear pores allow for the passage of macromolecules and ribosomal subunits in and out of molecules. So next, we have endoplasmic reticulum. This is connected to the nuclear envelope. The 3D network of the continuous tubules that course through the cytoplasm. We have two types of endoplasmic reticulum, the rough ER and the smooth ER. The rough ER synthesize or processes and sort proteins targeted to membranes, vacuoles, or secretory pathway. And in the smooth ER, these synthesize lipids and oils. Also, the, the endoplasmic reticulum act as an anchor point for actin filament control cytosolic concentration of calcium ions. So endoplasmic reticulum proteins are made in rough. So here in the endoplasmic reticulum, this is where proteins are being made. Lumen by an attached ribosome. Protein detached from ribosome, the ER fold in it on the ER fold in on itself from a transport vesicle. The transport vesicle buds off and moves to cytoplasm. It's either fuses with plasma membrane and fuses with Golgi apparatus. So the next one is the Golgi network, where the proteins or lipids made in the ER contained in a transport vesicle fused with the Golgi. The Golgi modifies proteins and lipids from the ER, sort them and package them into transport vesicle. The transport vesicle buds off and moves to cytoplasm, which fused with plasma membrane. So we have also the mitochondria. The mitochondria contain their own DNA and protein synthesizing machinery, ribosomes, transfer of RNAs, and nucleotides. Thought to have evolved from endosymbiotic bacteria. Divide by fusion the DNA in the form of cellular chromosomes like bacteria. The DNA replication is the interdependent from DNA replication in the nucleus. So the mitochondria is the site of cellular respiration. This process requires oxygen. This is composed of three stages, the glycolysis, the Krebs cycle, and electron transport. When we say glycolysis, this is glucose splitting occurs in the cell. Glucose is converted into pyruvate. And in the Krebs cycle, the electrons are removed, carriers are charged, and carbon oxide is produced. This occurs in mitochondrion. And last one is the electron transport. Electrons are transferred to oxygen. This is produced H2O and ATP occur in mitochondria. The next one is the chloroplast. It contains their own DNA and protein synthesizing machinery. Ribosomes transfers RNA nucleotide thought to have evolved from endosymbiotic bacteria. It divided by fusion. The DNA is in the form of cellular chromosomes like bacteria. DNA replication is interdependent from DNA replication in the molecules. Membranes contain chlorophyll, and its associated proteins are called the site of photosynthesis. They have inner and outer membranes, and the third membrane system is called thylakoids. Stock of thylakoids is known as granum, surrounded by the stroma, which work like mitochondria. During photosynthesis, ATP from stroma provide the energy for the production of sugar molecules. The vacuole can be 80 to 90% of the plant cell. They contain within a vacuolar membrane known as tonoplast. They contain water in organic ions, organic acid, sugar, enzymes, and secondary metabolites. They required for plant cell enlargement. The turgor or turgor pressure generated by vacuoles provides a structural rigidity needed to keep herbaceous plant upright. So the plant cell wall. A plant cell wall was first observed and named simply as a wall rather, by Robert Hooke in 1665. In 1804, Carl Rodolphi and J.H.F. Link proved that cells have interdependent cell walls. A cell wall is a structural layer that surrounds some types of cell situated outside the cell membrane. It can be tough, flexible, and rigid, which provides cell with both structural support and protein. There are two types of properties of cell wall, the rigidity and permeability. When we say rigidity, these are tensile strength, the hydraulic torture pressure, inflation, and in permeability, there are primary cell wall 30 to 60 kDa, carbon oxide or water, which is known for apoplastic flow and pH. The cell wall layers are the following. We have middle lamella, primary cell wall, and plasma membrane. So we have pectin, cross-linking glycan, and cellulose microfibres. The cell wall layer has a thickness of 0.1 ohms to several ohms. The layers are the following. The primary cell wall, which is thin, flexible, extensible, that contains cellulose, hemicellulose, and pectins. And the primary cell wall is also a permeable that size to 30 to 60 kDa. The secondary cell wall are thick, present inside the primary cell wall, contains lining for strength and density of wood cellulose fibers. And the middle lamella is the outer layer between the adjacent plant cells. This contains pectin. This gives stability or forming plasmodesmata. This made up of calcium and magnesium pectase. The plant cell wall are held together by middle lamella, made up of cellulose, cycloglucan, pectin, proteins, ca-ions, lining, and other ions, and water. The compositions of cell wall are primary cell wall and secondary cell wall. When we say primary cell wall, these are cellulose, hemicellulose, or cycloglycan, and pectin. Plant epidermis are cutin and wax, and the secondary cell wall has three: cellulose, which is 35 to 50%, cyclan, which is 20 to 35%, and lignin, which is 10 to 25%. The formation of middle lamella first formed from cell wall plate during cytokinesis, while in the primary cell wall, this is composed of cellulose fibrils produced at plasma membrane by cellulose synthase complex. Microfibrils held by hydrogen bond or tensile strength. Secondary cell wall constructed between plasma membrane and primary wall. Plasmodesmata interconnecting channels of cytoplasm that connects protoplast. Eukaryotic cell walls composed of polysaccharides known as chitin and polymer, which is the cellulose. Chitin and cellulose joined by beta 1 and 4 linkage. Example are fungal cell walls, algae, water, molds, slime molds, and others. Fungal cell wall consists of chitin and polysaccharides. The matrix of three components are chitin, glucans, and proteins. The algal cell wall consists of cellulose or glycoproteins. Components of it are mammans, silans, alginic acid, and sulfonated polysaccharides. In water mold, this consists of cellulose about 4-20% and glucans. The slime mold is composed of cellulose. The prokaryotic cell wall are known the example of prokaryotic is the bacteria. Bacteria cell wall major component is peptidoglycan, which is known as a strong shell. The gram-negative bacteria has a thin cell wall, and the gram-positive bacteria have a thick cell wall. Archaeal cell walls have a lack of peptidoglycan, composed of pseudopeptidoglycan sulfated polysaccharides and glycoproteins. The function of it is to give rigidity and strength, to act as a physical barrier to the plant, to prevent cell swelling and bursting as a result of osmotic pressure, and to promote cell-to-cell signaling through their cell walls. So now we will talk about the role of biochemistry in livestock. So in animal husbandry, the quality of milk can be checked by biochemical test. It is also help diagnose any diseases condition in animals and birds. In fisheries, the water quality is regularly monitored by biochemical tests. Any drastic change in water chemistry and composition of fishery ponds can lead to vast death of fishes and prawns. Hence, the tests are done on a regular basis to see salt content or the calcium content, the pH level, accumulation of waste due to not changing water for long, and others. And in adulteration, even the composition of food material produced, their alteration and adulteration, for example, in honey, can be found by biochemical tests. Biochemistry tests help prevent contamination. And enhanced yield in fisheries use of substance to promote fish growth and their reproduction and others. So biochemistry plays a valuable role in fishery, poultry, sericulture, beekeeping, and livestock and the others. So now, let us know the role of chemistry in food industry. So biochemistry enzymes used in food industry in the renate, lactase, and protease and catalysis. So here in the picture. So we will talk that one by one. In dairy industry, biochemistry has a role in rennet, lactase, protease, and catalase. In rennet, extracted from the fourth stomach of young calves, these contain enzymes that cause milk to become cheese. Excuse me. It separates solid curd and liquid. Different animals rennet are used for different cheese. Most common vegetable rennet is enzyme. In lactase, this is present in the brush border of the small intestine, artificially extracted from yeast, required for the digestion of whole milk used in production of lactose-free milk. Also used in production of ice cream and sweetened, flavored, and condensed milk enzymes. The catalase produced from bovine livers or microbial sources breaks down hydrogen peroxide to water and molecular oxygen. Along with glucose oxidase, it is used in treating food wrappers to prevent oxidation. Also used to remove traces of hydrogen peroxide in the process of cold sterilization. And in protists, this is widely distributed in biological world. Hydrolysis the specific peptide bond to generate para-K-casein and macropeptides in production of cheese. Results in bitter flavor to the cheese and also in desired texture. So now, let's jump to the baking industry. We will talk about maltogenic amylase, glucose oxidase, and pentosanase. In maltogenic amylase, these are the flour supplement. It has anti-stalling effect. It modifies starch while most of the starch starts to gelatinize. Resulting starch granules become more flexible during storage. Glucose oxidase are oxidized glucose and produce glyconic acid and hydrogen peroxide. H2O2 is the strong oxidizing agent that strengthens the disulfide and non-disulfide cross-link in gluten. Good working condition help proper function of bakery system. So this is the structure of a glucose oxidase. We have also the pentosanases. Pentosanases or pentosanases. This extract mechanism is not yet discovered. Improved though machinability, yielding more flexible, easier to handle dough. The dough is more stable and gives better oven spring during baking. Now, we will talk about the role of biochemistry in meat industry, and we have protease and papain. Protease cleaves the bond that hold the amino acid together. As the enzymes break apart proteins, which disrupts or loosen muscle fibers and tenderize it. In papain, found in papaya, 95% of meat tenderizers available in grocery stores are made from papain. It is extracted from latex in a papaya fruit. These enzymes are purified and sold in powder or liquid for enzymes. The role of biochemistry in paper industry are the following. Conventional way of making paper pulp, making pulp using enzymes or biopulping, pulp bleaching using enzymes or biobleaching, enzymes used for de-inking, GM trees with less of lining, and papers from bagasse. Correct me if I'm wrong. In mechanical pulping, the bark logs are forced through rotating tooth steel disc. The disc tear the logs and remove the lignin. The lignin degraded gives paper a brownish tingy, which are used for printing newspapers. Making pulp using enzymes or known as biopulping, it improves penetration and effectiveness of chemicals during the cooking of wood chips for separating the cellulose fibers from the lignin. Biopulping reduces the demand for energy and chemicals, improves paper quality, and decreases the environmental impact of pulp production, according to Pullman et al. In 1998. Biotechnology, silviculture, trees, and other bioresources can be used to enhance the properties required in cellulose fibers. The pulp bleaching using enzymes is known as biobleaching. Chlorine is used for bleaching process, which has a huge polluting potential. Pulp is usually tinged with brown color due to lignin content. Enzyme enhances this bleaching process by two ways. Silase breaks down the carbohydrate silane and to reduce the need for chlorine in bleaching, but gives number of byproducts dioxin and PCBs rather. Lipases are used to control deposit of pitch. Catalase is used to convert residual hydrogen peroxide to water and oxygen. Biobleaching of pulp with enzymes advantages are the following: the reduction of chlorine consumption, pulp dewatering, dinking, removal of pitch, degradation of dissolved and suspended organics in concentrated influence of mills. It enhances fibrillation to give stronger paper, according to Erickson in 1997. Biobleaching eliminates few of the processing steps, thereby simplify and reduce the severity of treatment of water waste. Enzymes used for de-inking. Cellulase enzymes are used for de-inking process. This makes the use of recycled paper as a variable option to reduce the number of trees needed to be cut to make a paper. The inking process involving sodium hydroxide, flocculants, dispersants, and superfactants is used widely currently. The alkali can make the treated pulp yellow, and subsequently hydrogen peroxide is used. The alkali can make the treated pulp yellow, and consequently hydrogen peroxide is used subsequently to bleach the alkali de-ink pulp. The GM trees with leaves of lignin. Lignin is a chain of galacturonic acid that binds the cellulosic fibers, can be reduced by making JM trees with less of lignin, but wood with less of lignin degrades quickly to release more carbon dioxide in the atmosphere. Paper from bagasse or bagasse through the deforestation is a matter of concern for both an environmentalist and the paper industry this has forced. So that's all for my topic. Thank you for listening.